Welcome. My name is Keduo Qian with the Division of Lifecycle API within Office of Pharmaceutical Quality. In this webinar, we will be discussing the regulatory considerations for synthetic and semi-synthetic oligosaccharide complex APIs in generic products. Synthetic and semi-synthetic oligosaccharides, for example, low molecular weight heparins or pentasone polysulfate sodiums, are a group of complex APIs with heterogeneous mixtures of oligosaccharide components that have a distribution of molecular weight or structures. In order to assist potential applicants prepare an abbreviated new drug application, the agency has published product-specific draft guidance on enoxaparin sodium, dalteparin sodium, and pentasone polysulfate sodium. In this webinar, we will present the agency's current thinking on the active ingredient sameness assessment of the oligosaccharide complex APIs in the generic and reference products. The topics such as the source material equivalence, the sample preparation, the critical process parameters and the critical quality attributes, primary characterization methods, as well as negative controls will be covered in this webinar. First, I want to very briefly introduce the low molecular weight heparins and the pentasones to those of you who are not very familiar with this type of complex APIs. Low molecular weight heparins are anticoagulants that are produced by depolymerization of unfractionated heparin. Heparin is a complex, naturally occurring polysaccharide found in certain animal species. Its backbone consists of repeating disaccharide building blocks. In comparison with the unfractionated heparin, low molecular weight heparin has the advantage of lower adverse event rate of heparin-induced thrombocytopenia, which can be fatal. Currently, 3 low molecular weight heparin products, enoxaparin sodium, dalteparin sodium, and tinzaparin sodium, have received U.S. Food and Drug Administration approval for the treatment of thrombosis. In this slide, the structure of enoxaparin sodium, dalteparin sodium, and tinzaparin sodium has been shown. You can see from the structure of the repeating disaccharide building blocks. Pentasone polysulfate sodium was approved in 1996. It is indicated for the relief of bladder pain or discomfort associated with interstitious cystitis. PPS is a semi-synthetic heparin-like macromolecule carbohydrate derivative. It chemically and structurally resembles glycosaminoglycans. It is prepared from naturally occurring xylem extracted from the beechwood tree. PPS has a molecular weight of 4,000 to 6,000 dalton. The structure of PPS is shown in the bottom of the slide. We can see that both low molecular weight heparin and PPS are complex mixtures, which means they do not have defined batch-to-batch consistent stoichiometry. In our review perspective, these complex mixtures are treated as a single active ingredient. Thus, to support an active ingredient sameness for these complex mixtures, the ANDA applicant should try to characterize each component in multiple lots of the mixture for both the proposed generic drug product and the reference product under similar conditions. The acceptance ranges should be proposed for each component in the active ingredient mixture with justification. Also, rationale for acceptability of the differences should be proposed. To facilitate the ANDA applicant develop the generic version of such complex mixtures, agency has published product-specific guidance. We can compare the product-specific guidance for low molecular weight heparin and PPS. For low molecular weight heparin, 5 criteria are recommended. First, you should demonstrate the equivalence of physical-chemical properties. Second, the equivalence of heparin source material and mode of depolymerization should be established. Third, equivalence in disaccharide building blocks, fragment mapping, and sequence of oligosaccharide spaces should be demonstrated. Fourth, equivalence in biological and biochemical assays should be demonstrated. And fifth, equivalence of in vivo pharmacodynamic profile should be shown. For PPS, we have 3 focuses in the review to establish the pharmaceutical equivalence. First, source of naturally occurring starting material. We should demonstrate the starting material used are the same. Second, concerning the physical-chemical properties, the molecular weight distribution should be comparable. The overall structure property of characteristic fingerprints, for example, the degree of sulfation, sodium content, Raman and IR spectra should be equivalent. And third, the equivalence of monosaccharide building block composition and chain branching. For example, the xylose units, sulfation pattern, the glucurotic acid groups, linkages, and anomic configurations should be shown to be equivalent. The ANDA applicants are recommended to develop a combination of validated low-resolution as well as high-resolution analytical methods in order to establish the sameness between their generic products and the RLD reference. Such studies include but are not limited to the listed below. For example, the weight average molecular weight and the distribution by gel permission chromatography, the stoichiometry studies, the UV, FDR, and Raman spectra should definitely be provided. The fingerprint profile of the saccharide chain should be investigated. The strong ion exchange HPLC and reverse phase ion pairing liquid chromatography are shown to be suitable in the analysis of such molecules. However, you are free to choose your own analytical methods as long as it shows enough resolution. Also, the chemical composition fingerprint analysis should be studied by proton NMR, carbon NMR, as well as suitable 2D NMR. In the following several slides, I would like to cover some of the issues we encountered during the review of oligosaccharide complex APIs, so that you understand what should be expected if you wish to submit such applications. First, for low molecular weight heparin products, for the sample comparison, we would request at minimum 3 commercial-scale batches to be compared with multiple reference-listed drug lots. The second, some applicants would provide more data comparison with other approved generic products. Please note that the agency cannot evaluate sameness comparison with other approved generic low molecular weight heparin. Per 505J and the citizen petition response, the demonstration of sameness can only be done using the reference-listed drugs. For low molecular weight heparin manufacturing, the applicant needs to follow the criterion number 2 to demonstrate the equivalent heparin source material, as well as the same mode of depolymerization. This is because the manufacturing details can impact the composition, as well as the molecular weight distribution of the final low molecular weight product. The resulting composition and content of the oligosaccharides are critical for the pharmaceutical equivalence to the reference list drug. Developmental studies should be carried out to correlate the critical process parameters to the critical quality attributes. The acceptance range for the CPP should be justified by the pharmaceutical equivalence criteria number 1 and number 3 studies. In addition to the USP requirements for the heparin sodium starting material, the agency would also require characterization of the building blocks because they have inherent direct impact on the final sameness of the low molecular weight heparin to the RLD. So you need to provide the test for major disaccharides and tetrasaccharides in the heparin sodium starting material specification. Also, you need to provide tests for the 1,6 anhydrous building blocks, the content trisaccharides, content of tetrasaccharides, including the linkage isomer. The establishment of the acceptance criteria based on the analysis of multiple batches of heparin sodium USP should be provided. As mentioned previously, a combination of low and high resolution techniques should be developed to establish the sameness. The high resolution method include but are not limited to chain mapping, disaccharide building block mapping, and fragment mapping by sex HPLC, for example, the composition by NMR. We want to provide the current accepted quantitative comparison criteria. Use enoxaparin as an example for fingerprint chromatography peaks that comprising more than 10% of the total peak area. We would accept the range of 90% of the RLD Lowinox minimum to 110% of Lowinox maximum. For peaks that are greater than 1% but less than 10%, agency would accept 75% of Lowinox minimum to 125% of Lowinox maximum. For peaks that are above the quantitation limit and to the 1%, a qualitative peak comparison would be okay. In the disaccharide building block study, a specific trisulfate delta IS should be controlled at the 95% of Lowinox minimum to 105% of the Lowinox maximum. So this is the current accepted criteria for the quantitation. To ensure the drug substance quality, selected primary characterization tests should be included in the drug substance release and stability specification as well. The use of negative controls in the method validation of the oligosaccharide complex APIs are unique because of the very complex nature of the mixtures. The negative control is needed because we need to demonstrate the specificity and the sensitivity of the characterization methods with respect to the structure fingerprint. So it is very important that the applicants develop sufficient negative controls to cover all the manufacturing process parameters which can potentially affect the structure fingerprints. The negative control are expected to meet or marginally pass the basic criteria of all the USP requirements. For example, the molecular weight distribution, the 1,6 anhydrous content, and the bile acid specified in the RLD labeling. However, the negative control should fail at least some of the primary sameness characterization tests. For example, the chain mapping, the chemical composition by NMR, the disaccharide building block mapping, etc. Thus, such negative control can capture the process significance of individual manufacturing. The negative control should be applied to all the sameness studies, and the results of the negative control should be compared to multiple reference list drug lots. For pentasone polysulfate sodium, we would also request at minimum 3 commercial scale batches to be compared with multiple reference list drugs. Different from the low molecular weight heparins, which are injectables, the PPI's drug product is actually a capsule containing the API PPS as well as excipients. Therefore, the extraction of PPS from RLD is necessary. Complete and clean recovery per the label claim of the PPS should be demonstrated so that the obtained analytical results can represent all the PPS components within the reference list drug. Because PPS is manufactured from the naturally occurring xylem, which are extracted from the beechwood tree, the natural raw material used for the manufacturing should be specified. The identification of the plant species should be up to the cultivar, so that the source material sameness can be established. The essential structure features of the API can be impacted by the natural source and the manufacturing mechanism. Thus, the manufacturing details and all the process parameters should be reported. The process developmental study on the correlation of the CPP ranges to the CQAs is required. Similar to the low molecular weight heparins, a combination of low and high resolution techniques needs to be developed to characterize the PPS structures. The data for the structure features such as the building blocks, the types and positions of the linkage and their orders, the branchings, for example, should be demonstrated and compared with the RLD. Just as low molecular weight heparins, the negative controls are necessary in the PPS study in order to demonstrate the specificity and sensitivity of the analytical methods with respect to the structure fingerprints. Sufficient negative control should cover all the manufacturing process parameters that could affect the structure fingerprints. And the negative control should be applied to all the tests and also to all the RLD lots. Here are some common deficiencies that we observed in assessing the oligosaccharide API applications. First, the manufacturing critical process parameter ranges are not justified by the critical quality attribute test. Second, the negative control batches were not developed properly, and many times the negative control batches do not cover all the CPP ranges that are identified. Third, many times there are insufficient data provided to establish the drug substance sameness. Fourth, the primary characterization methods are not validated properly. Here, we would not only request the validation of the drug substance stability and release specification test methods, we require all the primary characterization methods, including the fingerprint method, to be validated. And fifth, many times the drug substance specification does not include primary characterization fingerprint test. Such test is needed so that the drug substance quality is ensured. The agency recognized that development of the oligosaccharide complex APIs can be very challenging. That's why the prospective ANDA applicants are encouraged to contact us. You can submit the controlled correspondence or request a pre-ANDA meeting with the Office of Generic Drugs in order to discuss your proposed methods for demonstrating the sameness. The controlled correspondence is appropriate if an applicant has a specific and targeted inquiry about the generic drug development. A pre-ANDA meeting is appropriate for seeking a dialogue with the agency on a particular matter that would fall out of the scope of controlled correspondence. Information on the enhanced pathway for discussion between FDA and the applicant for complex product is available in the guidance for industry. Resources on controlled correspondence, on pre-ANDA meeting, product-specific guidance about the low molecular weight heparin development, and the Q&A are available in this page. Thank you for looking at our posters. After reviewing the webinar, please remember to submit the questions. Use the link provided on the workshop poster page. Questions received by February 15, 2021 will be included in the poster Q&A session on March the 3rd. Questions received by March 19 will be included in April 9 poster and presentation. Thank you.